Hello and welcome to my channel. So today we are going to briefly discuss B cells and T cells, how they functionally mature and how they perform their function. All blood cells which are formed in bone marrow are structurally and functionally mature except B and T lymphocytes, which are only structurally mature. For functional maturation, T lymphocytes go to thymus, present in the chest or mediastinum, while the B lymphocytes go to lymph nodes. So first of all, we are going to talk about the T lymphocytes. So when a new T lymphocyte is formed in the bone marrow, it goes to thymus for functional maturation. Now what the thymus does is that it differentiates the T cells into different types of T cells. One thing we must remember that T lymphocytes are not a product of thymus. They are actually a product of bone marrow, but they are functionally matured in the thymus. Now what does the thymus do is that thymus assigns different duties to these T cells. Now we must remember that all the T cells in our body have different cell surface receptors. And these cell surface receptors are called clusters of differentiation. Basically, these are the receptors through which we can differentiate between different types of T cells. So the first one is killer T cell. It has CD8 cell surface receptors. Normally it is a phagocyte and also antigen presenting cell. So for example, any foreign body like bacteria or virus infiltrates our body. The killer T cell catches this foreign body, engulfs it, and releases lysosomal enzymes and digests it. After the foreign body is destroyed, the killer T cell presents the antigen on its surface, and this is why it is called antigen presenting cell. Must remember that killer T cells, natural killer cells, and monocyte or macrophages are natural phagocytes and antigen presenting cells. The most important and most effective phagocytes and APCs are monocytes and macrophages. The second one is suppressor T cell. These cells suppress immune system not to attack our own body tissues. They help provide self-tolerance. The important point here is that one of the reasons of autoimmune diseases is defective suppressor T cell, or suppressor T cell is not working normally. So what it means is that if the suppressor T cell is defective, the immune cells would start attacking our own body tissues. For example, myasthenia gravis. It is a neuromuscular junction disorder in which antibodies produced by our own immune cells block, alter, or destroy acetylcholine receptors. This disorder is due to thymic dysfunction, for example, tumor, thymoma, hypertrophy, or atrophy, etc. As we all know that T cells are functionally matured in the thymus. So if the thymus is not working properly, then we get the autoimmune myasthenia gravis. Now we come to the helper T cell. It has CD4 cell surface receptors. The function of the helper T cell is that it helps the immune response. So for example, any bacteria or virus enters our body. What happens is that the macrophages catches them, engulfs them, and then digests them by releasing its lysosomal enzymes. Then the macrophage presents the antigen on its surface. The helper T cell is activated. It releases cytokines, specifically the interleukin-4, which activates the mature B lymphocytes. These B lymphocytes are then divided into two types of cells, the plasma cell and the memory cell. The memory cell retains memory of antigen, while the plasma cell starts production of antibodies. Namely, there are 5 antibodies in our body: the IgG, IgA, IgM, IgE, and IgD. The mnemonic for this is GAMT, by which you can remember it. The type of antibodies formed depends on the type of antigen. So, in a quick review, the macrophages catch foreign bodies, digests them, and presents the antigen on its surface to the helper T cell. The helper T cells release cytokines, which activates the mature B lymphocytes, and the mature B lymphocyte is divided into plasma cell and memory cell. The plasma cell then produces antibodies against the foreign bodies. Then we have the memory T cell. It retains response memory of antigen. Now we come to the B lymphocytes, how they functionally mature in the lymph nodes. As we know that lymph node also has outer cortex and inner medulla. The functional maturation of B lymphocytes takes place in the germinal cortex of lymph node. So when a new B lymphocyte is formed in the bone marrow, it goes to the lymph node. Then cracks appear in the small B lymphocyte and is converted into small cleaved B lymphocyte. The small cleaved B lymphocyte grows in size and is converted into large cleaved B lymphocyte. The large cleaved B lymphocyte then breaks down into smaller cells known as the small non-cleaved B lymphocytes. These cells then grow in size and are converted into large non-cleaved B lymphocytes. And these are the cells which mature into proper B lymphocytes. And with this, our topic for today comes to an end. If you like this video, kindly hit the like button, drop down a comment, share my video, and subscribe to my channel. Thank you.